In this video, we are going to discuss the preventive and the clinical trials, the two important components in research, in health and medical research, and both components are essential as I have said, in health and medical research, but they serve different purposes. But the two categories are required to enhance both the treatment and prevention of disease practice through generation of quality and evidence-based information. And when we talk about the clinical trials, that the overall aim of the clinical trials is focused on diagnosis, treatment, and disease management. And this include treatment trials like randomized clinical control trial, like, for example, diagnostic trials for testing a diagnostic kit for, or trials for the quality of life. And these trials concern evaluating the efficacy of new drugs, for examples, or other therapeutic procedures to test their efficacy, for example, surgical or any surgical intervention, physiotherapy, ionizing radiation, and, for example, a randomized clinical control trial to test a new chemotherapeutic agent for treatment of malignancy. The preventive trials, on the other hand, in general, the term preventive trials implies trials of primary preventive measures. And these trials are designed to prevent or eliminate disease on an experimental basis. And the most frequently occurring type of preventive trials are the trials for vaccines and chemo prophylaxis as main primary preventive measures that are used for prevention of the disease. Also, we have behavioral trials that test the lifestyle change, such as dietary change, such as exercises and sports and others, and as well as screening and early detection to assess the methods of early detection of disease. Here we can see the design of a randomized preventive control trial is that we have the reference population, which is the parent population for whom, if the trial has proved to be successful and the outcomes prove to be fruitful, then we are going to apply these outcomes on the reference population. And then we have to carry out some sort of random selection for the experimental population, which is actual population that participate in the trial. And then the experimental population is randomly allocated either to a group which received the vaccine and another group which do not receive the vaccine. And so the expected outcome among the group received the vaccine will be some of the group members will not develop the disease, other may develop the disease. And also among the group members who did not receive the vaccine, we expect that some of them will have the disease, others will not have the disease. This is the in general design of a randomized preventive trial, which is one of the important trials, as I have said, for testing primary preventive measures such as vaccines and chemoprophylaxis. Now we can talk about the phase of clinical trial. Before a new drug or an innovative medical approach can be tested on humans, extensive laboratory research is required and is carried, and sometimes may be spanning over several years or long duration in order to arrive to important outcomes. In most cases, this research involved conducting experiments on animals and or human tissues as well. And in case the studies prove to be successful, the investigator may send the data to an independent committee or technical task force that comprises of physicians, scientists, and who would then approve and monitor any further tests involving human participants. Then we move to phase two. The first step or phase one. The first step or phase one is to understand how well the intervention can be tolerated in a small number of individuals, and it does not meet the standard definition of a clinical trial as we have discussed before. And most phase one designs are relatively simple. Evaluating drugs needs first step is to estimate how a dose can be administered before unacceptable intoxication is experienced by patients or the health volunteers who receive the treatment. And this dose is usually called the maximally tolerated dose (MTD), and the number of participants usually ranges from 20 to 80 health volunteers. Then we go for phase two trials, and once the MTD is established, the next step is to evaluate whether the drug has any biological effect and to estimate the rate of adverse effect, if any, like side effects or any sort of interactions. And if the design of phase one has not been adequate, then the investigators may evaluate the maximum tolerating dose again to ensure if it is يعني, appropriate to proceed on. Phase two design depends on the quality of adequacy of phase one, and the number of participants is usually in the range of 100 to 300 participants. If the response rate is less than 20% among the participants, the drug is immediately omitted. If the response rate is greater than 20%, more patients are enrolled according to the precision desired, usually 10 to 20. Now we talk about phase three trials. These are clinical trials as defined, and phase three trials have a short period of evaluation and focus mainly on effectiveness, but knowledge on safety is also necessary to be generated. Larger group of participants are usually in either groups of participants are enrolled in ranging from 1,000 to 3,000 participants. Phase four trials, or what we call it post-marketing studies, delineate additional information including drug risk, benefits, and optimal use of the drug after being used on wide scale. Uh, Now we talk about registration and publish of clinical trial, registering and publishing clinical trials. Registering a clinical trial when it begins and making all the information related to the study publicly available has many benefits. And registration is usually mandatory for the journal publication. If you are planning to publish an article, then it's very important to register the clinical trial before starting the trial or with the beginning of the trial. Researcher can access the United States Library of Medicine website using the following address, which is shown here. To register a clinical trial, you must determine who is responsible for registering the study and which protocol registration and results system account should be used, and as well to understand the submission requirement, what is required, and to log in to the registration system and enter the required information, review, revise, and submit the records as well. This is important. The results obtained in clinical trials are often published in peer-reviewed journals, and when doing so, it's important to maintain statistical and reporting integrity of the published manuscripts, especially if they deal with research on human subjects. This can be achieved by checking the articles against common reporting guidelines, such as the Consolidated Standards of Reporting Trial, the consort statement, or, for example, the standards for the reporting of diagnostic accuracy studies start, or the strengthening the reporting of observational studies in epidemiology, STROBE, which is called the STROBE statement. These are very important reporting standards and frames that can help the investigator to finalize the outcomes of the trial. Thank you very much.